Thanks for the introduction, and thanks to the VanBug organizers for giving me the chance to present some of our work. So like Elodie said, in my lab we're focused on the application of genomics data to questions in the nervous system, and to do that we look a lot at gene expression data. We also have projects looking at neuroanatomy in relationship to gene expression. So we have interest in neuropsychiatric disorders, more basic issues. And the approaches that we've been taking, like Elodie mentioned, is to use lots of data sets, so meta-analysis. And we're interested in gene networks. And this is something that excited me from the beginning of my entry into the field of computational biology. And so the story I'm going to tell you about is maybe a little bit of it could be a little bit of a downer, sort of a cautionary tale. But it's also, I'm very excited about it because I think it's given us some insight into how maybe we can start to make some real progress in using gene networks. So, I mean, I gave the talk a little bit of a obnoxious title, and I added a subtitle on the way over for how to cope with the problems I'm going to talk about. So the outline of my talk is as follows. So I'm going to first sort of frame the issues and how gene networks are used, in particular in the context of gene function and how it's been scaled up in the post-genomic era. And I'm going to talk about these problems that the title refers to, which is all due to what we call gene multifunctionality. And so there's sort of two main stories and then a shorter one on the distorting effects of hubs in gene networks. Another is the distorting effects of critical edges, and I'll of course explain what I mean by that. And I'll mention briefly the broader impact on what people often like to call unbiased studies, where we think there's actually substantial bias that can be uncovered. So, so the central challenge that motivates a lot of the work in my lab is how do genes produce phenotypes, or how do genes function, or how do phenotypes get produced by genes, however way you want to look at it. So this is a picture of some fruit flies, which I like, but don't work on anymore. And, you know, the classic example, which is one of the first genetically identified phenotypes, which is the white phenotypes of the fly at the bottom there. So this is one of the first genes that was ever mapped, and this sort of sets the stage for, this is back in the early 20th century, for many decades of trying to figure out what genes do. So the question that we ask as biologists and as computational biologists is whether this is hard or not, and you get kind of two answers. So, so in one sense, we've kind of come to realize that gene function is not something that's easy to relate to phenotypes, because it turns out that there's not that many genes in the genome. There's not one gene for each phenotype. There's not enough genes to explain all the phenotypes. So a lot of traits or diseases are clearly very multifactorial interactions between the gene and the genes and the environment, but many genes. So any one gene may only have a small effect when you remove it, or it's so we think of things as being related to combinations of genes. On the other hand, it's easy because now we have technologies that let us analyze all the genes. So microarrays or sequencing methods are good examples. And now we can generate lists of candidates that are associated with a trait of interest or a disease quite easily. So again, for the past 10, 15 years, there's been increasingly this idea that, oh, well, you know, the genome is very complicated, but high-throughput methods are going to give us the answers. So I'm kind of casting my talk in the frame of function prediction, and I think that a lot of people, maybe in computational biology and bioinformatics, we're sort of familiar thinking about function prediction, but really this is a fundamental kind of task, and whether you call it function prediction or not. So I'm just going to give a couple of examples of what I mean, where function prediction may play a role. And again, taking it from a sort of computational point of view. So one you first think of is that there's a lot of genes that we just don't know what they do. So this is a figure that's, and a quote taken from the papers announcing the draft sequence of the human genome in Nature and Science back in 2001. And at that time, there was a huge swath of genes in the genome that really no one knew what they did. They may have had homology to genes in other organisms, but no one knew what those did either. So now, 10 years later, some gaps have been filled in, but there's still thousands of genes where the function isn't known. And of course, the question is, can we figure out what they do using computational methods? Often though we know what a gene does in some sense, but we have a phenotype and we don't know what genes underlie it. And so there's a problem of trying to assign new functions to genes. So one example is in, this is kind of a goofy figure, but it just reminds me what to say. Often in genetic studies, you may isolate a region of the genome that you think harbors a gene that's influencing the phenotype. So in analysis of copy number variation in the genome, this is a common situation where there may be a deletion of a segment of the chromosome that has in it 10 or 20 genes. And this is just an example of a tool that I pulled off of a paper. You can find lots of examples of this where people try to get some idea of what it is, which genes are in the region, of course, but then what do they do? Which one is the smoking gun? Which one is the causative gene for the phenotype? So there's a lot of computational methods that people have developed to do that. So we think of this as a prioritization task or a gene function prediction task where you're only focusing on the genes within that genomic interval. So that's a little bit more of a narrow problem than just saying what is the function of this gene. I don't know anything else about it. So another place where we often try to use what again can be broadly thought of as function prediction is when we're doing experiments that involve a manipulation of a biological system and gives us a readout which might be represented as a list of genes. So in this case, it's very similar to the genetic case, except now we have a ranking of genes based on, in this example, how differentially expressed they are in two conditions. And so again, people say, well, we can do this high-throughput study and get this list of genes, but then they're stuck with the problem of how do we interpret this list? So in this list of all these genes that I found, they're differentially expressed, which ones are really the important ones to focus on? Why these genes and why not some other genes are turning up? What are the commonalities between them? And so this is all the kind of question that has motivated a lot of computational work to try to develop methods that would help dissect this out and turn this list into an easy answer. What are these genes actually doing? Why did they go up in that condition? So those are three examples. So of course, determining gene function, I gave the fruit fly, the white gene example, determining gene function is not a new thing. So now at some point, we didn't have computational methods, and people actually had to determine gene function somehow. They actually had to go in the lab and do something. So how do they do that? So if you're not used to thinking about this, it's kind of like, hey, I wondered how did they actually do that? So back in the old days, and these are still things people do, of course, is that you would use genetic mapping to show which genes are influencing which phenotypes. And if you could narrow it down to a specific gene, then you would say the function of that gene is to produce that phenotype. So for the white gene, the function of that gene is to make fly eyes red. And then you just build up from there. So biochemistry is obviously a key tool where you isolate you isolate an activity. So you have a substrate that turns blue, and you find ways to run your samples through various columns and isolation methods, and see which find the part that makes it turn blue, and then you find the protein and you microsequence it, and then it says, aha, this is the gene that degrades lactose. And so then there's structural methods, so you can get the 3D crystal structure, for example, of a protein, mutate residues, and see which ones actually matter to the function. So all these methods are used to build up, in a very gradual way, information about what genes do. Now, of course, again, people do this, but increasingly, we rely on approaches that build on this information. So we're not just doing an experiment, we're using prior knowledge. So a classic example, a classic approach is what we just call inference, where we have a gene A and we have another gene B. And so maybe say we know something about gene A, but we don't know something, we have a question about gene B. So if we have some way of linking these two genes together or associating them, there. I know it's high tech. So we might say, well, maybe this tells us that there's some relationship between the function, something about gene B, that we can learn from gene A. So this is a quote from a from the high-throughput era, from a biochemist who was very excited about the advent of these approaches that were being applied, just beginning to be applied in sort of genome-wide manner, that discovering a protein of unknown function interacts with one of known function provides a valuable clue. So here's an actual example of this that comes from before the high-throughput era. And I think this is a kind of cool example because I think maybe not many people know about this. So APOE is a gene which, if you have a certain allele of it, your risk for developing Alzheimer's disease when you get old is dramatically increased. And it was originally identified as when people were trying to find proteins that bind to beta amyloid. Beta amyloid is a protein that exists in the plaques that develop in the brains of people with Alzheimer's disease. That was just discovered with biochemistry. People were looking for proteins that bind to it. And someone had said, oh, this protein over here binds to that. They were trying to test that hypothesis, and they couldn't reproduce the result, but instead they had a contaminant. And this contaminant turned out to be APOE. So beta amyloid binds to APOE. They would call this that there's some kind of guilt associated with APOE. So APOE is implicated in Alzheimer's disease by this binding. And then in the same paper, which was published in 1993, they show that there's actually genetic variants that are associated with increased risk. So that's one type of association, protein binding. There's obviously a lot of different kinds of associations that you can imagine, so which are based on the kind of things that, again, people start with when they're trying to determine the function of a gene. So what else resembles its structure, its primary or secondary, tertiary structure? What other genes have the same mutant phenotype? What other genes have mutations which enhance or suppress that phenotype? What genes are expressed in the same pattern? What proteins bind to its product? I said that already. And what genes have similar patterns of conservation? So these are all types of associations or guilt that you'll see in the literature. There are probably others that I've forgotten about. And of course, biologists have been using these principles for many years, like the APOE is from 1993, before the genome. If it works for one gene, of course, you might wonder, well, does it work for many genes? So then, now, fast forward to the current era, you cannot open a journal without getting hit in the face with a network. So, or if you're at a conference and walking down, whatever that's about. So you'll see lots of these kinds of pictures. So there's the idea that because genes on their own have small effects, that their function is somehow represented in a network. So it's like systems biology all of a sudden. So network analysis attempts to make use of this sort of distributed nature of phenotype-genotype relationships, sort of to build on this sort of association between genes giving us ideas about their function, and scaling up what biologists have always done one gene at a time. So again, we call this idea guilt by association as a generic term. It's really just saying this, making an inference based on the properties of two genes that you can sort of transfer information about one gene to another. But the basic idea that's been used in computational biology a lot for the past, and biologists, a lot of tools that biologists use now to analyze their high-throughput data use this kind of idea. So use networks or data that can be kind of thought as a network to predict function or prioritize candidates. And again, there's all these different types of ways of defining the links between genes. So in the common paradigm for actual function prediction, where you want to make, you're actually using like a machine learning method to predict function, to evaluate your method, you're going to use cross-validation. So in any case, you're always going to have there's usually some idea of a training set. So we have genes that we know have a function, so like they're involved in Alzheimer's disease, and then we look in some sense nearby them in the network to see what genes are there, and we would infer that those genes are also involved in Alzheimer's disease, for example. Okay, so this is a very popular approach. There's many, many, many papers that have used it. This is one example, which I think is from a year or two ago, from the work of Edward Marcotte, where they were making gene networks to do prediction of function in Arabidopsis. So the figures kind of doesn't matter, the details don't matter, but basically what they're showing up here is how they start with known and unknown genes, and they transfer labels to genes that they want to guess their function, and you generate these kind of RSE curves, which I'm guessing most people are familiar with. And what they sort of say in all these papers is that, hey, we can predict gene function, and everything looks pretty good, so let's go ahead and apply this. And there's lots of sort of cases where people have done this successfully. So, so here's a few quotes from papers like this where people have sort of said that these methods work really well. The bottom one is maybe a good summary. Computational approaches quickly provide accurate, unbiased predictions of protein function. So, so I guess the rest of the talk is going to be about. I probably could have written one of these quotes at some point. But when we were trying to apply these methods we were particularly interested in genes in the nervous system, so we were trying to pick genes that might be involved in synaptic plasticity, for example, in the brain. And we found that the results looked kind of funny. I won't go into the details of what I mean by funny, but there was reasons to be suspicious. And so the rest of the talk is really about the uncovering of what's going on. Why those results, they weren't negative, they were too good to be true. And I'm not going to talk about the specific case, but about a general problem. So there's a huge gap in our knowledge of gene function. So there's a lot of interest in exploiting, finding function and then exploiting that information to learn new things about genes. The fundamental approach that people use is guilt by association, if they're not starting de novo. Again, people have, like those quotes show, is that there's many cases where people have shown that the computational methods can be used to scale up this kind of analysis to genome-wide. So again, or have they? So please interrupt me if you have any burning questions, but I'm going to go now into this thing about multifunctionality. So it turns out that a key property of genes that influences the way we analyze their function is how many functions they have. And what I mean by that is how many functions that we know they have already or have been annotated. So a multifunctional gene is simply a gene that has multiple functions. We have, now you say, well, how do you define that? Because you don't know. But we have a working definition, which I think is pretty well justified, which is the number of gene ontology terms. It's actually a slightly more complicated measure, but that's basically, first approximation, that's what you can use. The gene ontology is this one way of describing function, and I've sort of challenged people to come up with a better alternative, and it turns out that this has a huge amount of explanatory power for the phenomena I'm going to discuss. So I acknowledge up front that defining gene function is kind of difficult, and maybe get into a philosophical argument about it, but operationally, this is what biologists do. They use things like the gene ontology terms. You can replace this with KEGG pathways or whatever. It doesn't really matter. The gene ontology is the biggest and arguably the most used. So importantly, multifunctionality is a term that maybe people get confused about. It's not the same thing as pleiotropy or hubbiness in networks or promiscuity. So some people say a promiscuous protein, one that binds lots of things. So that's not what we're saying. We're just saying genes that have a lot of different functions defined by their gene ontology terms. So now I'm going to show that there's a relationship to things like hubness that turns out to play a very important role. But we're not, so I've seen some people say, oh, a hub that defines multifunctionality in a network. So if you don't know what a hub is, you will in a few minutes. Okay, so a kind of fundamental question is whether multifunctional genes are interesting. And the argument I'm going to make is that in a sense they're not, because by definition they're involved in lots of things. So if you're interested in one thing, then that gene can't be specifically related to that. So if you're interested in cancer and you find a gene that's involved in cancer and 200 other things, well, that gene is not specifically, it may be important. So the prototypical examples, well, you'll see some in a minute. You can probably guess what some of the genes might be that have lots of functions, but because they're so multifunctional, they're not specifically related to what you're doing. So again, it's sort of maybe an unusual way of thinking about it. Usually people think of genes that are critical and multifunctional as the most interesting genes. And I'll develop the argument a little bit more by the end of the talk. So what it turns out is that because that algorithms that we use in computational biology tend to focus attention on these genes. And the reasons for that I'll explain. And that turns out to be a problem if you think those genes are sort of generic. If you're interested in making a statement about schizophrenia, then you would like to find genes, I argue, that are specifically as possible involved in schizophrenia and not just doing everything. So here's the first indication of a potential problem. So actually, I'll go back a couple slides here. Whoa. So remember I showed these RSC curves here. So this is so the area under this curve is commonly used as a measure of learning performance. So it's the false positive rate plotted against the true positive rate. So you want the curve to be up near the upper left. So what they've done here is, and this is a good example, is because they have two sets. Forget the overlapping curves, but they've done separate learning tasks for these two different classes of functions they're interested in. So here they're interested in abiotic response, and they get an RSC of about 0.8, the area under the curve. For organ development, they do it again. If you're familiar with the RSC curves, these should look pretty bizarre to you, but let's not get into that. But they get, and again, about 0.7 or 0.8. So that's sort of the basic paradigm. It's like for all these different categories of genes, they're going to train the machine learning algorithm, which could be very complicated, do cross-validation, get an ROC score back out, and do that for all of the groups. Okay, so now, what I'm going to show you in this, what's in this figure here, is showing that you can, that if you want to, so say your targets are Go groups. So you have, so in this case, a whole bunch of, thousands of Go terms that you want to learn. Can you come up with a single list of genes that will allow you to predict the membership of genes in any Go group? So I'm going to walk through this carefully because it's a pretty important concept. So what we've done here is that we've ranked the genes by their multifunctionality. So we have a single list of genes, and so here I forget which genome this is. I think it's human. So we're taking all the human genes that we have any information about. We rank them by how many gene ontology terms they have, and then for any gene ontology term you want to guess, we simply return that list of genes. So you want to make a prediction about abiotic response. Here's this list of genes with the candidates at the top. Simply, the gene at the top is the one with the most Go terms. And the reason that works, that you might think it might work, is that if you want to guess which gene is in any given function, you might as well guess the gene that's in the most functions. So when you ask people how well you think this might do, remember that in real life, these groups are learnable. People report 0.7, 0.8, 0.9 to be crazy high for most gene ontology terms. This is the distribution you get. Pretty much every single gene ontology term is incredibly predictable simply by ranking the genes by how many gene ontology terms they have. So there's no algorithm here. There's no training. We're just ranking the genes by how many Go terms they have. So this outperforms any living algorithm out there. And you're probably, maybe some of you are thinking like, wait a minute, you're using Go and then predicting Go. And you're right, so it's circular. But it shows a potential problem. What if, what if your data actually ranks the genes by how many Go terms they have and you didn't know that? Okay, so here's some of the heavy hitters, the genes that have lots of Go terms, and probably everyone in the room who's done any biology can recognize a few friends in here. This is just like there's more. I just didn't want to put too many on the slide, but you'll see, P53, I think it has like 800 Go terms. So no matter what you're studying, simply guessing that one of these genes is involved will actually get you pretty far. You don't need to do any experiments, just write the frickin' grant about P53. You're probably right. Okay, no matter what it is. Okay, abiotic response, sure, probably. Organ development, yep. Any one of these genes, maybe guess two to make sure you cover all the Go groups. So that's how I get my grants. So, all right, there was laughing, so you kind of get the idea. This is like an important concept that these genes that have lots of functions, they're interesting and important, but if you just want to use those genes as as your starting point, really you'd like to find something a little bit more focused. Okay, so again, I raised the point of, does real data somehow encode this ranking of genes? So this is, again, is important. So if it does, if real data just spits out a list of genes ranked by their Go terms, number of Go terms, and you're doing cross-validation based on things like Go terms, then your function prediction will seem to work just fine. Your cross-validation results might look pretty good, but it's not going to be actually very useful because your predictions can be replaced by just ranking the genes by how many Go terms they have. That's not specific to your function. So again, any experiment that sort of encodes this list will seem to have interesting results, but it's just going to tend to assign functions to genes that already have them, so this sort of rich get richer effect. So then when you go and try to do, like, make new predictions, well, you have no real evidence that you've actually learned anything if this is the underlying problem. So I wouldn't be telling you all this if it didn't actually have an effect. So I'm going to walk through a short, hopefully, case study where, a real case where this plays a role. So this is a collaboration that my lab has with Ivica Separovic, who's a professor in pathology, and she's interested in the genetics of intellectual disability. And so she's been studying copy number variation in individuals with intellectual disability. And the task that they were trying to do was to identify this kind of prioritization approach, which genes in the C and V copy number varying regions in these individuals are most likely responsible for the intellectual disability phenotype. So because there's a long list of genes, you could go look at them by hand, but what they did was they tried a bunch of different candidate prioritization programs, which are all based on this gene guilt by association approach. So the inputs are the genes that are covered by the C and V region, an input of a training list of genes, so a list of genes that are known to play a role in intellectual disability. And our contribution to this was basically to suggest that they try a control where they take randomly selected genes as the training set, or genes involved in some other disorder like breast cancer, which presumably has little connection to intellectual disability. So the other input is a gene network, which is provided by the program, and again, they all work in this basic way. And the output is going to be a ranking of the genes in the C and V region by their priority. So the result is that the experiment and the controls yield similar rankings of genes. So whether you use a random set of training genes or breast cancer genes versus intellectual disability, the algorithm just spits out the same list of genes. Of the genes in the C and V region, it likes the same ones no matter what function you're asking about. Okay, when I say no matter what, I mean we tried a couple, right? So here's an example of this, this deletion on chromosome 19. There were 97 genes. The top candidates when you train the algorithm with known intellectual disability genes are these genes. And if you just train with random genes, you get 4 out of the same 5 genes back. So, okay, so that's a phenomenon, and but we've more recently shown that this is actually strongly correlated with multifunctionality. So, so again, I've said that in guilt by association, what will happen is that the genes that are best annotated are going to tend to get ranked the highest. And again, the reasons for that I'll get into a little bit. So again, it doesn't mean that the predictions are wrong, but they're too generic. So this plot shows the multifunctionality of the genes is quite strongly correlated with how they're prioritized by the algorithms. So that seems to be the explanation for, so basically, if your C and V region has any gene that has a lot of functions, the algorithms are going to tend to get stuck on them. So now why is that? Why do they get stuck on them? So the answer, what part of the answer is that the genes that have lots of functions tend to have lots of network neighbors in these networks. So they tend to be hubs. So this is a phenomenon that people have reported in the literature. There's been some equivocation, but it definitely seems to be true that. So this is a somewhat smooth plot of genes versus their multifunctionality plotted against node degree, so how many connections they have. So the hubs are over here, and there's definitely a correlation. So why does this matter? Well, apparently the algorithms get sort of sucked into the hubs. So the hubs are connected to everything. So if your gene is connected to a hub, it'll transfer the function from your test gene from the hub to your test gene, and you'll be right according to the cross-validation because of the multifunctionality of the hub. So what happens? So here's a more rigorous demonstration of this, where we've used a sophisticated machine learning algorithm plotted here. These are the RSC scores for the algorithm on this axis. On the x-axis is the RSCs that we get for using just ranking the genes by node degree. So now we're not ranking them by how many Go terms they have. We're actually using a collapsed version of this gene network where we're just ranking them by how many connections they have. So it's no longer circular. At each point is a Go term. And so there's obviously a correlation. The real algorithm does a little bit better, but not by much. And what's really important here is that there's a correlation. So the Go groups that are really learnable, they get a really amazing RSC score using the Gene Mania algorithm, are exactly the same ones that do really well by node degree. So this is pretty strong evidence that that algorithm is really being heavily influenced by node degree. So basically, there's about half the performance of the algorithm in general. We've done this for many networks. We've tried other algorithms. We've tried different species and so forth. It seems to be a general phenomenon. So protein interaction networks, co-expression networks, and so forth. So about half of the performance is accounted for by this effect. So you can see that here, the real algorithm does do better. So what's going on? Is there actually still something salvageable in these data? So the next part of the talk is about another problem that also has to do with multifunctionality, but it shows itself in a slightly different way. So I have to define a couple of terms, and actually, really, for my purposes, only one of them is all that important. And hopefully this is not one of the slides. I think this is the part of the talk I did on the bus. So we might hit a rough spot or two with duplicate slides or something. So a functional edge in a gene network is one that connects two functionally related genes that share a function. So that's what we want to know. We want to know. Ideally, our network would be entirely made up of that, but we don't have that information necessarily. So a critical edge is a network where if we remove it from the network, our ability to predict function decreases. So that's a definition. So and the intuition is that why we would want to look for these things is that we don't really want all the predictability of a function to come from one edge. So ideally, we would have lots of functional edges, and so therefore none of them would really be critical for any given function. Hopefully this is making some sense. Yeah, so here's the bus effect. So I already defined this. So right, so the question is, how common are such critical edges? Do they actually exist? And then the deeper problem is what effect this has on the usefulness of guilt by association analysis. Okay, so the next definition is of an exceptional edge. So an exceptional edge is one that's critical for predicting many Go groups or functions. And intuitively, if two genes are both highly multifunctional and share a lot of functions, which they will if they're both highly multifunctional, and an edge joins them, then that edge is more likely to be critical for many functions, because the way that guilt by association works is by looking at edges that connect genes that have the same function, fundamentally. So we predicted that exceptional edges would be the ones that join the most multifunctional genes. So you can develop a simple approach for choosing which is the most exceptional edge. Okay, so we started out looking at this question in a large, very well described set of gene networks for mouse. So this was used in a sort of friendly competition of gene function prediction algorithms that was published a few years ago. So this is the sort of lead paper that sort of summarizes the whole thing, and this sort of special issue of Genobiology that was dedicated to all the papers. And they gave the participants 10 networks of different types. There was a co-expression network, there was a protein domain network, there was a conservation network, and so on. And so for basically all the mouse genes, and when you collapse these all together, you have a network with 4.5 million edges. And they gave the competitors some Go terms to work with, which had this many genes. And then so what we're doing is sort of replicating some of what they did and evaluating the results with precision recall curves, where instead of RSC, now we're plotting precision versus recall. And what this does is, again, precision is you have high precision if you're in your ranking of your guesses. If your first one is right, then your precision is starting off well. And then recall is how long down your list you have to go before you get all of your true positives. So it's sensitive to whether your very first ranked gene is correct. So again, there were 9 competitors in this competition, and the algorithms all basically did very similarly. But my point isn't about the competition; it's about we use this network. So what we did is we showed that. So what we did is using the algorithm I described, where you just simply take the edge between the genes that have the largest number of Go terms in common, so what we predict to be the most exceptional edge, and just build a network from those, starting from the most and keep going. So for any genes, we're not using the real network here. So remember, this whole network the real network has 4.5 million edges. So we have a network of 23 edges. That's not 23,000 or something. It's 23 edges that gets the same level of performance as the full network. So now we've cherry-picked these edges. Here they are. And actually, if we keep adding edges, we can do way, way better than the real network does. Okay, so again, we've cherry-picked these edges. It turns out that a lot of these edges are actually in the real network. And you can predict the performance of any one of these 10 substituent networks. You can predict how well they do at learning Go groups on average by how many of those edges they have. So out of the top most exceptional edges that we predict, this is a plot of the average performance versus how many of those connections they actually have. So the networks that don't have many of them don't do very well. The network that has a lot of them, 2 networks that have a lot of them, are the highest performers. So that suggests that maybe a lot of the performance that they're reporting using precision recall is coming from a very small number of edges in the network. So which should be kind of scary, that you can take a network of 4.5 million edges, throw out 99.99% of it, and still do okay. So now we've done some more exhaustive testing, and I'll probably be going through this a little bit quickly. But what we did, so that network is really big. So we worked with some yeast networks, which are very high quality and again have been used a lot in the literature. And so what we did is we exhaustively tested the criticality of edges. So instead of guessing which ones are going to be critical or exceptional ahead of time, we said, let's actually determine this experimentally. And you do this by removing one edge, do the entire experiment, all the Go groups on that new network that's missing one edge, and see how your performance changes for all those Go groups, and do it again for the next edge. So this network has about 72,000 edges. So we're testing 72,000 different networks across 1,000 different Go groups. So this is an example of the kind of thing that we get. So in the inset here, so this is showing our precision recall measure of performance, and the distribution is of the number of edges which get that performance. So most edges in the network, when you remove, so here's the true performance, with the unperturbed network. If you remove an edge, it most of the time doesn't matter. But there is one edge that when you remove it, the performance goes way down. So this is for one example Go group. And this is the sub-network in the protein interaction network we're using for this particular Go group. The genes, the black squares are the genes that are in the Go group. The gray squares are genes that are connected to those genes. The genes that aren't connected aren't shown. They might be connected to the gray genes. And then, so there's actually just a few edges that are between the genes within the Go group. And it turns out that this edge over here, this is the critical edge. If you remove this edge, performance drops dramatically. So it's this one gene here that's connected by itself to this gene here that's not so isolated. That this gene here was the one that you can predict very nicely based on its connection to this gene. It has no other connections. And so that's a problem, because when you're doing cross-validation, all you can really make a statement about is your ability to predict this one gene. It turns out that none of the other genes have that property. You're not going to be able to make new predictions based on this trained algorithm. So in other words, we have no basis for saying that even though the performance might be quite good, we might decide it's good enough to make predictions, there's nothing about these other gray genes here that would lead us to even vaguely believe that they should be given the same confidence in their prediction as this one gene. This gene is an outlier. So okay, that's one Go group. How many Go groups did you have to look through, Paul, to find that one? Well, it turns out that most Go groups are like that, and in most networks. So, so this is 6 different networks. I'll spare you the details of what they are, but there's a genetic interaction network, there's protein interaction networks, and this is the number of Go groups that have, and the fraction of their performance that is accounted for by one connection. And obviously, there's a lot of Go groups that are impacted strongly by one connection. So sort of the outlier here is this network here of genetic interactions, and it turns out that this network doesn't provide that much learnable information in the first place. So not too surprisingly, if you have nothing, if you don't have very good performance in the first place, then there's not much to lose. Okay, so this is a very common phenomenon across all these Go groups and across all these networks. So we can also show this another way by, say, taking the edges. So one thing it turns out, I forget if I have this on the slide here, is, yeah, I guess I do. So if we take the most critical edges, so we can decide which of the edges are critical based on some threshold for as many Go groups as possible, and measure, as we remove those Go groups, how does performance drop. So this is the average performance across Go groups as we remove the gray curve just 200, 400 edges. Remember, the original network has many, many thousands of edges. So just removing the small number of edges hurts performance quite quickly. Similarly, if we build a network that's based just on exceptional edges, we quite quickly surpass the performance of the real network. And if we just randomly build a network using real edges, but randomly picking them, you can see the performance doesn't climb very fast at all. So because it's also, and those are averages, but if you look at the distribution of precision for all the Go groups that's shown here, if you use just exceptional edges, most of the Go groups tend to be learnable. So in other words, there's a lot of Go groups are sharing the same edges, okay, so they account for a lot. And if you remove the exceptional edges, then performance drops for a whole lot of Go groups. Now there is a little bit of an exception here, and that's, I think I took these slides out, but basically there are a few Go groups that are still learnable, and those are protein complexes. So protein complexes tend to be very highly interconnected in the protein interaction networks, so they don't have critical edges, but they're kind of unusual. There's a small number of them, and their properties are not, you can't predict new members of the complex using this information, and they don't really tell you anything about the learnability of other Go groups. Okay, so what this is telling us is that if you're using cross-validation performance to decide whether the network contains learnable information about gene function, and therefore you could go on and make new predictions, you can't do it. All the information that you're getting is coming from a tiny number of edges, maybe a few hundred edges in these big networks. And if it wasn't for those edges, you'd have nothing to learn. And those edges don't let you generalize to make new predictions. Okay, here's the protein complexes. Let me skip that since I'm getting kind of late here. So let me skip this too. So it's kind of, I mean, you can ask me about it if you want, but basically we have some ideas about ways to identify which edges. I mean, it's kind of an important point that we can predict which edges are likely to be exceptional without using information on their effect on performance, just looking at how many connections the genes are connected to are. So hubs can afford to lose edges. So if we privilege, all right, so so the higher the node degree of the gene, the less likely that its edges are going to be critical. So you can remove edges from hubs and still retain performance. Okay, so the last thing I want to talk about just very briefly is that, okay, like all this stuff about gene networks, maybe you're not interested in gene networks, maybe you don't think you're interested in function prediction, and I've tried to twist, some things into being function prediction that maybe you say aren't, like when you have a gene list and you want to categorize the genes on that list and do gene function enrichment analysis. Okay, so just the very brief story is that this is dramatically affected by gene multifunctionality. So this is a very commonly used approach. So most people have probably heard of things like GSEA. And one thing that the GSEA folks did at the Broad is they set up this database called Multifunctionality. That's a database of differentially expressed genes. So they've gone through lots of papers and pulled out the gene list, the hit list that those authors provided. And they have 3,000 of those lists. So this is a nice resource for us to use. And we asked the question, first, do those lists tend to be multifunctional? And the answer is yes. So genes that are commonly differentially expressed, according to Multifunctionality, tend to be multifunctional. that kind of makes intuitive sense. It just gives another measure of the same thing, that if a gene is showing changes in expression under lots and lots of different conditions, it tends to have lots of gene ontology terms as well. It's just sort of popping up in these methods, in these assays. Okay, so then here's the last, I think the last data slide, where this is just one part of the story where now to show that if you do, so then you take these lists of genes, 3,000 MultiDB lists, and you can do gene set enrichment, enrichment analysis on those lists. The authors probably did something like that, and they would have said, oh, this list is enriched for ubiquitin something or other, and so on. So what happens if you remove one gene out of that list? Okay, so out of that hit list. So now, you'd like your results to be robust, but we have an idea of which gene is going to be the most important to pick, and that's the most multifunctional gene. So what we did is we took all those different MultiDB lists and removed the most multifunctional gene, and then see how much of an effect does it have on the list of Go groups you get back out. And it turns out that a lot of the time, you get a huge change in the results. Removing one gene completely scrambles the Go groups you get back out. So that's these guys out here. This is the change in the rankings of the Go groups that started out as the top 10, the sum of their ranks. So they go down on average like 800 ranks. Now for this group over here, they don't change very much at all when you remove one gene. And it turns out that almost every case, these lists are themselves incredibly enriched for multifunctional genes in the first place. So they end up giving you lots of Go results back out, but they're all due to these multifunctional genes, and they tend to bring up lots and lots of Go categories because of that. Okay, so just to sum up, so one thing I'd like to leave you with is the question of what is it that makes a gene interesting? So I mentioned this at the beginning. So you might be able to find a gene that's obviously related to your favorite process, again, just by guessing P53, but the gene you pick might be find might be kind of generic. And I think that's what people have to kind of come to grips with, is that the genes that are going to be more specifically involved in their biological process of interest are probably going to be more subtle, and they're going to be harder to find. So that's a challenge. So the second point is whether, okay, I've dissed networks a lot. Are they still useful? So, I think, of course they are. You just have to be aware of these sort of gotchas. So one is that the assumption that function is a sort of distributed property in the networks, we don't have very good documentation for that. It's based on things like cross-validation of learnability of Go groups, and that turns out to be very misleading. That analyses of networks are so heavily influenced by the effects of hubs that hubs sound interesting, but they turn out to be sort of a vacuum, where things get sucked into. If your gene of interest is connected to a hub, and you want to say something about, if you think that's interesting, it's not. Every other gene is connected to that hub as well. So think about it. So of course, there's still local information in the network. So just knowing that gene A binds to gene B is still useful information, but you have to be careful not to let these other problems mess you up. Okay, so what to do about it, you can sort of be aware of it. So again, you can test to see whether your gene ranking is associated with multifunctionality. If you look on our website, we have some lists of genes. I didn't put the URL up here, but if you send me an email, I can point you to that, or it's in the paper as well, the one out which is in the slides. And then also to start to adapt our approaches so that the predictions we make for function, we want them to be specific for that function. So we should be testing lots of functions and picking the predictions that aren't necessarily the strongest predictions, but the ones that are most specific to, so if a gene that's predicted to only have one function, even though it's kind of weak, might be better than a gene that's strongly predicted to have lots of functions. Ideally, we would use data that's function specific, so most gene networks are not function specific to one condition or something like that, so you ideally would have data that's specific to the context, and that you have a control case that lets you contrast your two conditions. And then if you have multifunctional genes in your training set, so like your intellectual disability genes in my example, like you don't want those multifunctional genes to be in there. Okay, so to quickly conclude, so that multifunctionality is lurking as a confound in the interpretation of many biological experiments. It has a serious effect on computational attempts to exploit known information about gene function, especially in the context of what we think of as gene networks. And correcting for it is pretty challenging because multifunctional genes are important, hubs are important, removing them wholesale may not be the best idea. So I have to give, this is the most important slide, the acknowledgments. So pretty much this whole story grew out of, again, the curiosity we had a few years ago about why the gene function prediction results we were getting looked so good, but we didn't think they were right. And Jesse Gillis, who's a postdoc in my lab, and he's here, was really the one who he ferreted out what was happening and went on to develop this whole big story about how multifunctionality screws up all these different kinds of analyses. So he received some help along the way from Ella Mercier, who's a PhD student in the lab, Raymond Lim, who recently graduated with an MSc, and there were some other people in the lab who contributed in various ways, especially at the early stages, in struggling to do predictions and before we realized what was going on. So Vineet, Gavin, Anna Maria, and Fong. And I want to thank Avitsa and her colleagues for the collaboration that we've had on copy nerve variation in intellectual disability and our funding. And there's a couple citations there for papers that are relevant to this, though not all of it's published. So I don't know if we have time for questions, but I can take them over beer. They're multifunctional.
But the transcription factors might be involved in many different functions. So in this case, did you test?
So I actually don't know. I don't have a great answer to that. So kind of, but we've done things like if you take, like there's 3 Go trees, right? And you take just the molecular function hierarchy versus the biological process hierarchy, it doesn't really matter. So I mean, they're multifunctional, so I doubt there's any simple pattern like that. I don't know, maybe Jesse has some insight into that, but.
If you try to use a traditional gene set enrichment tool, you give a list of genes.
Right.
You ask which functions, after multiple test correction, is it enriched for?
All of them.
It would be enriched for 3,250 out of 3,268. So which of those functions is the interesting one? Is it enriched for transcription? Yes. Is it enriched for the brain? Yes. The ordering of that list will primarily depend upon how small and therefore subject to like seeding statistical significance that functional list of genes is. So if it's a big list of genes, it's not going to appear at the very top because it can't be, or it might be, if it's biased heavily, it will be more likely to appear as being significantly enriched. So it will be property of the set. It will not be the property of
one gene.
Yeah, it will have nothing much to do with the function in a weird way.
Right.
That's my.
Yep. Steph?
Wondering the same thing. So because we're using a number of gene ontology terms as a measure of functionality, would things turn out any differently if you use some other measure of functionality? And
Multifunctionality?
For
no.
example, manually annotated the entire genome. And is there any influence of like the level of annotation of gene and how well studied it is?
Well, first of all, we get the same result if you just replace this whole thing with KEGG. So others.
That's still things that are well characterized.
Well, right. So okay. So maybe what you're getting at is, our genes that have lots of Go terms, are they really multifunctional, or are they just are they better characterized? So genes that are obviously annotated at shallow levels in Go will just not have as many Go terms. It's the ones that are deeply annotated that will tend to have lots of Go terms. But we've done all kinds of things like, played with the number of, the size of the Go groups. It doesn't really matter. I mean, it's a very robust effect. So, you can, again, if you can choose your gene organization scheme of choice. Evan?
Do you see any problem with the time aggregation of the network or the ontology? Like a lot of these functions at a given point in time may be mutually exclusive.
Given time, like in the cell, you mean? Right, so that's what I meant at the end. Exactly, that's a great point. So that ideally, like the networks themselves tend to be sort of static. At least the protein interaction networks that we work with, that they're not really considering, like under different conditions, which functions are relevant. So yes, ideally you would want your networks to be condition specific. And so one thing we're interested in that we've been doing is that with co-expression data, with microarray data, because that's collected under a certain condition, you can construct condition-specific networks. The problem is that co-expression networks are noisier than protein interaction networks, but if you aggregate enough data together, you actually get a very high-quality network. So the limitation is you need a lot of data. But yes, it's absolutely that. Ideally, if you had a network that was constructed only under the conditions where your function was relevant, that you'd expect that to give you better, better information. But again, most people don't do that. They use these protein interaction networks and ingenuity networks, whatever, and don't worry about the conditions because they aren't condition specific. Okay, everyone's thirsty. I'm thirsty. Thank you.
Done here in the